Welcome back, everyone. It's Dr. Linder. In this next this lecture, we'll be talking about the neuromuscular junction. So muscle cells are stimulated to contract, right? Muscles have the ability of contract, which means to generate tension as a result of neurons. Those specific neurons are called somatic motor neurons. We have motor neurons and then we have sensory neurons. Motor neurons, motor is going to go to muscles. Sensory allows you to feel and sense things. So we have somatic motor neurons. So the neurons that make contact at the neuromuscular junction. So they call it the NMJ, neuromuscular junction. So right here, we're taking a close look at an axon. And we know that axons here we see as the somatic motor neuron. But then we have axon collaterals. Here's an axon collateral. Here's an axon collateral. These means they're just branches off of the main somatic motor neuron, okay, or off the axon. If we follow the axon to the end, we get an axon terminal. At the very, very end of the axon terminal are what we call synaptic end bulbs. Over here too, synaptic end bulbs. Now, what we have at the very end in these synaptic end bulbs are neurotransmitters. The specific neurotransmitter that we need to focus on is called ACH, which stands for acetylcholine. Acetylcholine is always an excitatory neurotransmitter, meaning it's always going to make muscles contract. Acetylcholine. It is an excitatory neurotransmitter. Okay, so let's take a look and focus on a few other words I want you to focus on. We've already mentioned myofibrils, which are muscle fibers. You're going to hear this term down here at the bottom, sarcoplasmic reticulum. It's that blue webbing-like, that meshwork. Really important structure because that is responsible for storing calcium. That's where calcium is stored in the sarcoplasmic reticulum. We have a T-tubule. And a T-tubule, the T stands for transverse. Transverse tubule because it runs transversely to the sarcoplasmic reticulum and to all these muscle fibers underneath. And you can see those muscle fibers underneath the sarcoplasmic reticulum. Looks like some are blue, some are red. Those are the actin and myosin filaments. Those are the contractile proteins, actin and myosin. And I also want you to see how we have the sarcoplasmic reticulum on both sides of the T-tubule, right? So we could see that here's the transverse tubule, and then you notice how we have the sarcoplasmic reticulum on both sides butting up against it. So we have 1, 2, 3 structures associated. You got a T-tubule, and then at least one dilated part of the SR on this side, another dilated part of the SR on this side. Those 3 structures is what is typically referred to as a triad. A triad. Okay, so transverse tubule, sarcoplasmic reticulum, myofibrils. Notice we have lots of mitochondria here, right? This is a mitochondria, all of these throughout are mitochondria. Okay, so now we're going to be looking at the neuromuscular junction. This is where we really want you to focus and pay close attention. So here is the axon, and this looks like it's myelinated, right? That's a Schwann cell that's myelinating this part of the axon. We're getting right to the end here. We see that there's an axon terminal. And at the very, very end of the axon terminal, we see a synaptic end bulb. And what did I say is stored in that synaptic end bulb? We said a neurotransmitter called acetylcholine, ACH, acetylcholine. Now, where the neuron attaches to the muscle, you'll hear this word motor end plate. That's just the specific part of the muscle for which the neuron will attach to the muscle at the motor end plate. That's the attachment site. Okay, now we're going to take this section right here. We're going to blow this up. So now we're going to look to the right of it. And again, we have the axon terminal. This is carrying the nerve impulse. You can see the arrow coming all the way down, and it's going to send that impulse into the synaptic end bulb. And what did I say we have stored in the synaptic end bulb? Well, we have these synaptic vesicles, all of these little circles. These are little vacuoles or little saccules that contain a neurotransmitter. We can see it right here. A synaptic vesicle containing ACH, that excitatory neurotransmitter. It's really, really important. Now, where does the axon terminal or this end bulb attach? Well, we said it attaches to the muscle, to the specific part called a motor end plate. So right here, we could also see the motor end plate. Now, between the axon, the synaptic end bulb, and the motor end plate, we see that there is this space, this yellow gap between the nerve and the muscle. That space is called the synaptic cleft. That gap or that space is called a synaptic cleft. It's just the space between the axon terminal or the synaptic end bulb and the muscle or the motor end plate. Now, what we see a little bit happening right here and right here, we see that those synaptic vesicles are opening up, and it looks like they're releasing the acetylcholine. Well, how are they doing that? Well, they're doing it with the help of this, calcium. Calcium is influxing. That word influx means it's moving in to the end bulb. And this calcium is a messenger. And this calcium messenger is going to come in, send a relay message to the synaptic end bulb and say, hey guys, need you to release that neurotransmitter acetylcholine. And it does. That method by which it releases that neurotransmitter is what we call exocytosis. Exocytosis, a word that we already learned in about week 1 when we learned about the cell and the organelles. Okay, so now let's take a look at this section right here. What's happening at that space in the synaptic cleft between the axon terminal or the synaptic end bulb and the motor end plate. So let's look at it here. So right here, where it's number 1, it says acetylcholine is released from the synaptic vesicle. So we see that happening here, and we see it happening here. So those little red pyramids that, triangles, whatever shape they are, that are being released Again, it's being released by this method called exocytosis. And what is stimulating the release? Calcium, right there, the influx of calcium. So we have acetylcholine, it is being released, and we see it floating in the synaptic cleft, that gap between the nerve and the motor end plate. And what is this acetylcholine trying to do? The acetylcholine is looking to bind to, number 2, an acetylcholine receptor. And those green things, those green doorways, are acetylcholine receptors, aka they're called sodium channels. So you see, when it binds, the door opens up, and what rushes in? Sodium. And when sodium rushes in, it creates this excitation or an action potential. Okay? This invagination right here, that indentation or invagination, that is called a T-tubule, transverse tubule. Okay, so here is the axon, or I should say a neuron, and the neuron has these little dendrites, it has a cell body, and it has an axon. And it looks like it's myelinated to speed up the conduction. These are little Schwann cells. And the action potential gets all the way down to the end here. So here's this little box where we have the neuromuscular junction, the NMJ. The end of the axon attaches to the muscle at the motor end plate. So now, right here is the synaptic end bulb, where you have all these little dots. These little dots are the synaptic vesicles that contain acetylcholine. Here is the motor end plate. And this gap is the synaptic cleft. So the acetylcholine is released into the synaptic cleft. It's going to bind to those acetylcholine receptors or sodium channels. The sodium channel opens, sodium rushes in, creating excitation or an action potential, and it goes right down the T tubule. Now remember, here's the T tubule. The action potential is traveling down the transverse tubule. And what does the transverse tubule articulate with? The sarcoplasmic reticulum, the blue structures. And what's stored in that sarcoplasmic reticulum is mineral calcium. Well, let's see what happens. So we have this excitation going down the transverse tubule. It butts up against this structure here, and that's called the sarcoplasmic reticulum. And the sarcoplasmic reticulum stores calcium, and it's going to release that calcium. It's going to release the calcium, and it's going to have a major effect, right? When this sarcoplasmic reticulum here releases the calcium, what structures are deep to that? Well, the actin and myosin filaments, those thick and thin myofibrils. So somehow calcium is going to have some major effect on the thick and thin filaments. The thin is actin and the thicker one is myosin, and somehow that calcium is going to have a major effect causing a muscle contraction. Okay, and that's what I need you to understand, and I'm going to go over what exactly happens with calcium and this thin and thick filament of the sarcomere. And when I come back, I'm going to get into the actin and myosin fibers and how muscle contraction take place.